Warm greetings to all the fourth semester botany students. I'm Dr. K.Y. Pratibha and I would like to talk on microsporogenesis and formation of microspore tetrads in this session. So let us take up the basic question. What is microsporogenesis? The formation of microspores or pollen grains inside the microsporangia of an anther in angiosperm is called as microsporogenesis. A diploid cell in the sporangium called as pollen mother cell undergoes meiosis and gives rise to 4 haploid microspores. The sporogenous cells divide meiotically to form haploid microspore tetrads. The microspores in tetrads separates and become pollen grains. It is by meiotic division the diploid sporophytic cells give rise to haploid gametophytes. So if you see this picture, you will understand the meaning of microsporogenesis. The diploid pollen mother cell undergoes meiosis, that is meiosis 1 and 2, to form 4 microspores or pollen tetrads. Let us take up the microsporogenesis in detail. When the anther develops, the sporogenous tissue is formed by the contribution of primary sporogenous layer. So we have studied earlier that the archisporial cells will form primary parietal layer and primary sporogenous layer, and the primary sporogenous layer will form the sporogenous tissue inside the pollen sac. The cells of the sporogenous tissues functions as microspore mother cell MMC or pollen mother cell PMC and undergoes meiotic divisions to form microspore tetrad. So microsporogenesis is nothing but the formation of microspores from a pollen mother cell through meiosis is called as microsporogenesis. The microspores get arranged in a cluster of 4 cells and hence are called as microspore tetrad. As the anthers mature, they dehydrate and the microspores dissociate from each other and develop into pollen grains. In each microsporangium, thus we will see thousands of pollen grains which are formed and they are released with the dehiscence of the anther. So, dear students, microsporogenesis takes place inside the sporogenous tissue, and the sporogenous tissues will undergo meiosis and they will form the microspores. So let us take up the functions of the sporogenous tissues in detail. The cells of the sporogenous tissue have dense cytoplasm, nucleus, nucleolus and few small vacuoles. They are polygonal in outline and attached to one another. Microspore mother cell is also called as microsporocyte or meiocytes. The primary sporogenous tissue formed by the archisporial cells, either directly it can function as spore mother cell or it can divide mitotically into number of cells which then ultimately may become spore mother cell or microspore mother cells. So remember that microspore mother cells or pollen mother cells are diploid in nature. Microspore mother cell or PMC. The microspore mother cells are polygonal in shape and it is tightly packed until the anther begins to increase in size. So the pollen mother cells, they also start expanding. All the pollen mother cells can form the pollen grains, but some of them will disintegrate and act as a nourishment to the developing pollen grains. The plasmodesmata is the interconnection between the pollen mother cells or microsporocytes which are connected to one another and with the tapetum. You have to remember that the pollen mother cells are connected to one another through plasmodesmata and also they are connected with the nutritive tissue tapetum through plasmodesmata. But following the entry of the pollen mother cells into the meiosis, the plasmodesmatal bridges with the tapetum will be snapped or broken. So have to remember that when pollen mother cell starts entering into meiosis, the plasmodesmatal connection with the tapetum will be snapped. After the discontinuity of the plasmodesmata connection among the microsporocytes, so another point, now they have interconnection with each other, that is pollen mother cells are interconnected with the plasmodesmata. Instead of plasmodesmata, now cytoplasmic bridge, a bigger bridge or the channel will be formed. It is now let us come to the pollen mother cell. Pollen mother cell is covered by a primary wall made up of cellulose. But shortly before meiosis, this wall disintegrates and it will be replaced by a massive deposit of callose, which is nothing but beta-1,3-glucan outside the plasma membrane. So three things are happening. You have to remember in the pollen mother cell. The first thing is that it is increasing in its volume. Second thing is that plasmodesmatic connection with the tapetum is broken, and among the connection among the pollen mother cells will be broadened to form the cytoplasmic bridge. And the third thing is the PMC will be having the cell wall made up of cellulose, which will be broken. Instead of that, the callose wall, a new wall layer which is made up of callose, that is beta-1,3-glucan, is adding in between the place between the cellulose and the plasma membrane. Callose wall will start appearing. Callose deposition starts at the corners of the cells between the plasma membrane and the original wall. The deposition of the callose is initially incomplete, leaving many gaps through which there is an establishment of massive cytoplasmic channels between the microsporocytes. This is what I told you earlier. Cytoplasmic bridges or channels are seen between the PMCs which are undergoing meiosis. So this was mistaken before as cytomixis, but now they know that it is a cytoplasmic channel between the developing between the pollen mother cells which are undergoing meiosis. The cytoplasmic continuities of the PMCs impose a mutual influence of one cell over the other. All the PMCs undergo same meiotic division because of the syncytium. So this cytoplasmic bridge or channel help in the functioning of the pollen mother cells to undergo the same meiotic stage. This help in maintaining close synchrony during meiosis in the large number of PMCs in an anther locule. So there will be a synchrony in the PMCs in the anther locule. At the end of meiotic prophase, the callose walls of the PMCs close up. So the cytoplasmic channels, I told you that pollen mother cells will be having cytoplasmic connection with the neighboring pollen mother cells. So after the prophase 1, these channels will also be cut off because of the callose deposition. Therefore, additional callose walls are formed between the daughter cells of the PMCs. So let us take up another important point. These callose wall of the pollen tetrad or the microspore tetrads will be broken by the callase enzyme of the tapetum after the end of meiosis. So let us take up meiosis in pollen mother cells. Meiosis in Greek means lessening. It is a special type of cell division of germ cells in sexually reproducing organisms to produce the gametes such as sperms or egg cells. It involves 2 rounds of division that ultimately result in 4 cells with only 1 copy of each chromosome. So meiosis will reduce the chromosome number. It is a reductive. It is a meiosis is a reduction division where the diploid cells will give rise to 4 daughter cells which become haploid. Later on, during fertilization, the haploid cells produced by meiosis from a male and female will fuse to create a cell with the 2 copies of each chromosome again. That is, diploid condition is again formed in zygote. So meiosis is a reduction division. It is a type of cell division in which the daughter cells receive one half of the original set of chromosome of the parental cell. Hence, it is also called reduction division. Meiosis occurs only in germinal cells. Remember that in plants, only in the anther, inside the anther, the pollen mother cells can only undergo meiosis. Even in gynecium, inside the ovule, the meiosis takes place to produce the embryo sac or the female gametophyte. So it is cell division includes meiosis occurs only in germinal cells, that is in spore mother cells of the plants. Cell division includes two important processes. They are karyokinesis, division of the nucleus, and cytokinesis, division of the cytoplasm, resulting in 2 to 4 daughter cells. Karyokinesis is the division of cell nucleus resulting in 2 daughter nuclei, followed by cytokinesis in the second process, which starts after karyokinesis and the division of the cytoplasm takes place. So in cell division, we know that there is karyokinesis and cytokinesis. Karyokinesis is the division of the nucleus, whereas cytokinesis is the division of the cytoplasm. This takes place depending on the type of the plant. So let us go to the next one. Meiosis in pollen mother cells. Meiosis has two phases, as all of us know that, meiosis 1 and meiosis 2. In meiosis 1, there are prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis. While in meiosis 2, there are prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis. So I told you pollen mother cell will start undergoing meiosis, that is meiosis 1 and meiosis 2. In meiosis 1, prophase 1, there is leptotene, zygotene, pachytene, diplotene and diakinesis, which will be followed by meiosis 2. Meiosis 1 is the reduction division. Meiosis 2 is nothing but mitosis. So if you see this picture, you will understand the various events happening in meiosis of the pollen mother cells. In prophase 1, the chromosomes condense and the nuclear envelope breaks down. Crossing over occurs. In metaphase 1, the pairs of homologous chromosome move to the equator of the cell. In anaphase 1, the homologous chromosome move to the opposite poles of the cell. In telophase 1, the chromosome gather at the poles of the cells. The cytoplasm divides in successive type. In prophase 2, a new spindle forms around the chromosomes. In metaphase 2, the chromosomes line up at the equator. In anaphase 2, the centromeres divide and chromatids move to the opposite poles of the cells. In telophase 2 and cytokinesis, a nuclear envelope forms around each set of the chromosome and the cytoplasm divides. So this is the photograph of the meiosis happening in the fritillaria type of the plant, and in lily plant you can see all the different stages of meiosis, meiosis 1, meiosis 2 in a beautiful way. So let us take up the cytokinesis. In the meiosis, karyokinesis happens first, followed by the cytokinesis. Cytokinesis can be classified into two types. Microsporocytes after meiotic division undergo cytokinesis by any two process, simultaneous and successive type. If you see this picture, you will understand. In the successive type, after two nuclei has been formed, immediately the cell will form the cytoplasm also divide immediately after the first karyokinesis, and then the second karyokinesis takes place, and then the second cytokinesis, that is the cell wall is laid down. Whereas in the simultaneous type, the first karyokinesis takes place, two nuclei are formed. So this two nuclei will undergo further karyokinesis to form four nuclei. Only after four nuclei only after the formation of four nuclei, the cell wall is being laid in case of simultaneous type of cytokinesis. So let us take up the simultaneous type of cytokinesis. In this case, the first meiotic division is not accompanied by wall formation. As a result, after meiosis 1, a binucleate cell is formed. The two haploid nuclei undergo the second meiotic division synchronously by the virtue of their common cytoplasm. Later, callose wall are laid down as centripetally growing furrows which meet in the center of the cell to produce a tetrad. Successive type of cytokinesis. Here, immediately after the first meiotic division, wall is laid down centrifugally to form a dyad. The cell plate is formed in the center that extends laterally. It is followed by the deposition of callose on either side of the plate. The cells of the dyad undergo second meiotic division followed by callose wall formation in the same as the first division, thus resulting in tetrad. So, tetrads are formed after the meiosis. What is the meaning of the microspore tetrads? After meiosis, the four microspores formed, four daughter cells, which are nothing but microspores, are held together by a common wall of callose. So, they are bound within a callose wall. So, we call that group of the microspores which are together because of the meiosis as microspore tetrads. Microspore tetrads are formed, remain associated with each other for some time. The group of microspore is called microspore tetrad. The four microspores separate from each other. The tapetum plays a very important role here, which will release callace to break the wall of the microspore tetrad. And each of the microspore develops a characteristic shape or form which differs in different species of plants. After separation from tetrads, microspore is called as pollen grain or male gametophyte, which starts forming its own exine and intine wall layers. So, based on the shape of the tetrad in angiosperm, we will come across around 5 to 6 types of tetrads which can be seen in angiosperm. Angiosperms, tetrahedral tetrad. Here the most common type, 3 spores in one plane and 1 spore in other plane seen in Drosera. Isobilateral tetrad, all the 4 spores are formed in one plane, that is Siamese. In decussate tetrad, out of the 2 lower spores only one is visible, both the upper ones are clear. Example, magnolia, T-shaped tetrad, 2 cells present side by side and the lower cell forms 2 cells lying one above the other. Example, Aristolochia, linear tetrad, all the 4 spores are present one above the other in a linear fashion. Example, Halophila. Rhomboidal tetrad, spores are arranged in the form of rhomboid. Example, Annona muricata. Irrespective of the type of tetrad, eventually the wall of the tetrad, that is callose, is broken down and all the microspores are released freely in the anther locule, which will be floating in the tapetal fluid, and there the pollen grain starts to form its cell wall, exine and intine, and it increases and then it starts forming the male gametophyte, giving rise to vegetative cell and generative cell. So if you see this picture, you will know the types of microspore tetrad. A, tetrahedral tetrad. B, isobilateral tetrad. C, decussate tetrad. D, T-shaped tetrad. And E is a linear tetrad. So let us revise what we have done in this session. Microsporogenesis comprises the events which lead to the formation of haploid unicellular microspores. During microsporogenesis, the diploid sporogenous cells differentiate as microsporocytes or pollen mother cells, which divide by meiosis to form 4 haploid microspores. The arrangement of pollen grain in a tetrad is due to the specific type of cytokinesis. The cytokinesis can be of 2 types: simultaneous type, where the cytokinesis occur only at the end of meiosis 2. The resultant tetrad shows tetrahedral arrangement which is common in dicotyledons. Successive type, in this type The cytokinesis occurs twice, once at the end of meiosis 1, forming 2 cells, and then again at the end of meiosis 2 to form 4 cells. The resultant tetrad shows isobilateral arrangement and is found in monocotyledons. There are 5 types of tetrad: tetrahedral, isobilateral, decussate, T-shaped, and linear. Among these, tetrahedral tetrad is the most common type. Thank you.